Hi, I'm Alabia, and my talk will be focusing on a recently published work along with colleagues from Hokkaido University and University of Alaska Fairbanks. Here we looked at the patterns of marine biodiversity under a warming and sea ice-free Pacific Arctic. Climate change is known to drive species distributional changes. These range shifts are characterized by latitudinal and depth-related migrations. For instance, from the 1980s to present, demersal and pelagic species along the U.S. coast have shown poleward movement and migration into the deeper waters. More so, in the climate-sensitive Arctic region, climate-driven shifts resulted to the restructuring of marine communities. In the Barents Sea, for example, community structure between the cold and warm years showed contrasting patterns. In cold year, warm affinity taxa have shown a more southern distribution, while in warm year, their distribution expanded further north, resulting to borealization of its Arctic community. In the Pacific Arctic region, our earlier study have also shown that the distributional shifts between the Arctic and boreal taxa have differing rates of range shifts, which are dependent upon prevailing climatic regimes in the eastern Bering Sea and species' relative sensitivity to climate. For instance, the transition from a cold to warm climate showed a general lag in observed and expected species distributional responses, and that in general, Arctic taxa were better able to track climatic shifts than the subarctic species. Building on these reported contemporary patterns of climate-driven species and community shifts in the Arctic region, we extended our analysis to examine the impacts of future warming and sea ice loss to species distributions and how these anticipated range shifts affect the marine community structure and biodiversity distribution in the Pacific Arctic. In doing so, we expect to draw insights on how these changes will impact ecosystem function, particularly in the Arctic region. Our study area is shown in this figure, and the spatial extent is represented by the purple-colored polygon. So based on the existing literature, this study area can be further divided into distinct biogeographic and topographic domains. Our analysis primarily relied on the long-term species occurrence data from the annual summer trawl surveys conducted by NOAA, with sampling stations shown in this figure. The green points correspond to available sampling surveys in the North Bering Sea and Chukchi Sea. In the present analysis, these data were not included in the model fitting but were only used to inform the selection of pseudo-absences. We focus our analysis on the 20 major fish and 6 invertebrate taxa from the NOAA Summer Bottom Trawl Survey, which were consistently identified during the 25-year period between 1993 and 2017. The species pool is further comprised of a mixture of boreal and Arctic species. The table lists the climate and environmental data used for the construction of species distribution models. So these factors were significant predictors of species distribution based on our earlier analysis. So to obtain a final resolution climate data for future projections, we did delta downscaling by taking the difference between the present and future temperature and sea ice concentration data from the multi-ensemble mean of general circulation models under each RCP scenarios and adding these differences to the final result of satellite data. Now, using this data, we constructed species distribution models using 10 algorithms to generate species-specific distributions for present and future period. The model outputs for each species were then combined into a consensus mean for both present and future projections. Using the projected species distributions between present and future periods, we then generated species matrices to analyze taxonomic and functional diversity. Here, we looked at alpha and beta diversity for both taxonomic and functional aspects, as biodiversity is not only comprised of the sum of the species, but of their ecological or functional variety. While alpha diversity looks at the total number of species, beta diversity, however, examines the changes in the community composition based on species identity. From here on, I will only be showing the results between the present and late century period under contrasting warming scenarios, which registered pronounced biodiversity changes relative to the early and mid-century periods. So present-day species richness is primarily high throughout the eastern Bering Sea, with the lowest richness registered in the Chukchi Sea. This contrasts with the future projected taxonomic richness, which captures increases in species counts in the Chukchi Sea as the climatic habitat conditions become favorable for boreal taxa, especially under the RCP 8.5 scenario. Further, a notable decrease in species richness in the southwest part of the basin under RCP 8.5 scenario appeared to be driven by the northward shifts in the distributional ranges of both boreal and Arctic taxa. Here, representative species of boreal taxa in the outer domain are red king crab and red sole. Now, if we look at the functional richness, we can also see clear changes in spatial patterns between present and future periods. Present-day and late-century functional richness under the low warming scenario showed considerably higher functional richness relative to RCP 8.5, where the most prominent decline in functional richness was observed. This suggests that the future marine communities will be represented by species that are functionally similar and that the available niche spaces in the ecosystem are not fully utilized. So to further examine the present and future functional biogeographic patterns, we will now look at the species characterization based on 9 functional traits, 5 of which are continuous variables and the rest are categorical. The table shows the results of principal coordinate analysis based on the functional distance. Here, the 2 major axes accounted for a total of 85% of the variance in the species functional traits, with PC1 associated with species morphology and life history, while PC2 is associated with the differences in species mobility and resource utilization. Now, based on the major PC axis of functional traits, we also did spatial characterization of the study area based on the community-weighted mean of functional traits of the species found at each pixel. So the figures show the spatial characterization using the first principal component axis for present and future periods. In the eastern Bering Sea, the present-day functional biogeography highlights the dominance of larger and longer-lived taxa, while the Chukchi Sea were characterized by species that are smaller and short-lived. So this is particularly true for RCP 2.6. In contrast, the future functional biogeography in the Arctic region under the highest warming scenario, which is RCP 8.5, showed a profound shift in a community dominated by larger and longer-lived species. Here, large-bodied animals have body sizes greater than or equal to 49 cm and lifespan of greater than or equal to 17.5 years. Moreover, the future functional biogeography in the Arctic waters also showed a shift from less mobile to highly mobile and predatory taxa relative to the present and RCP 2.6 scenarios. This anticipated movement of large and predatory species into the Chukchi Sea will impact the future Arctic ecosystem function and structure, as the trophic transfer would tend to be faster and the predation pressure on the smaller Arctic species would increase. Likewise, the projected range expansions of boreal taxa under warmer and sea ice-free conditions in the Pacific Arctic will also make this system more vulnerable to climatic disturbances as trophic components become more connected. So, to summarize, our analysis revealed profound changes in the Pacific Arctic marine communities, which were projected from conditions of warming and sea ice loss. In particular, the future biodiversity redistribution featured an intense borealization of the Pacific Arctic communities, which are primarily driven by the influx of boreal taxa to the warmer and sea ice-free Arctic. This in turn drives the anticipated community shift in the future. Finally, the borealization signals could alter the Arctic ecosystem properties, which will increase its vulnerability as biological components become increasingly connected, allowing the cascades of environmental and climatic disturbances down the food web. Finally, I would like to acknowledge the funding agencies for this work, which were the Japan's Arctic Challenge for Sustainability Research Programs, as well as Arctic Research Center Gaizan Yukio project funded by Hokkaido University. Thank you for your attention.